Chapter 7. Growth Control. As we learned in Chapter 1, microorganisms are vital links in our planet's web of life. We couldn't live without them, but they do make us sick. They spoil our food. Sepsis refers to microbial contamination. Asepsis is the absence of significant contamination. Aseptic surgery techniques prevent microbial contamination of wounds. Sterilization is the removal of all microbial life, killing or removal of all microorganisms in a material or an object. When properly carried out, sterilization procedures ensure that even highly resistant bacterial endospores are killed. The method used is heat under pressure. Example is the autoclave. Disinfection is the removal of pathogen. It's the reduction of the number of pathogenic microorganisms to the point where they pose no danger of disease. Disinfectants are typically applied to inanimate objects, so things that are fomites or do not move. So example of this would be bleach. Antisepsis is the removal of pathogens from external living tissue. It's a treatment directed at living tissue. Antiseptics are chemical agents that can be safely used to destroy microorganisms or to inhibit their growth. Example of this would be iodine or neosporin or something. Degerming is the removal of microbes from a limited area. So that's when we wipe with alcohol, use an alcohol pad before we inject with a needle. Sanitization lowers microbial counts in eating utensils. It lowers the number of microorganisms in eating utensils and dishes to meet the public health standards. Sanitization may simply refer to thorough washing with only soap and detergent. Example of this would be a dishwasher. Chemical agents can have the following effects. Biocide or germicide means that it kills microbes, so usually it'll kill the bacteria. Bacteriostatic inhibits bacterial growth, so it's not killing the microbes. Just like how they grow exponentially, bacterial populations also die at a constant logarithmic rate, so they also die exponentially. So you'll see that being shown here in the table. There are factors that have an effect on the death rate of microorganisms. The number of microbes. The more microbes you have, the more time it will take to get rid of the entire population. So if we look at this picture here, we'll see over time the number of cells. If we have a higher population load, it's going to take much longer to kill them versus using a low population load. Low population load will die much faster. Environmental influence such as organic matter and temperature or biofilms. Length of exposure, how long chemical agents are being used on the microbes. Microbial characteristics, so the characteristics of the microbes, both chemical and physical. Actions of microbial control agents. The plasma membrane contains proteins, so that means it can be altered by alcohols. Membranes also contain lipids, which can be disrupted by substances that dissolve lipids. So here we can use alcohol or detergents to disrupt the lipids. So the consequences would be that there's a loss of a permeability barrier, so antimicrobial agents can reach. Damage to proteins. When proteins are altered or denatured, the functional shape of the protein molecule is destroyed. Any agent that denatures proteins prevents them from carrying out their normal function. Metabolism and transport is disrupted. Damage to nucleic acids. Nucleic acids cannot produce enzymes or make a new membrane. Physical methods of microbial control. Heat. The thermal dead point is the lowest temperature at which all the cells in a culture are killed in 10 minutes. So it's the temperature that kills all bacteria in a culture in 10 minutes. So it's the lowest temperature that I would need to kill them, all of the cells, in 10 minutes. Thermal dead point is the lowest temperature at which all cells in a culture are killed. Okay, I just told you the thermal dead point, my apologies. The thermal dead time is the minimal time to kill all the cells in a culture. So the time required to kill all the bacteria in a particular culture at a specified time. So thermal dead time is the minimal time to kill all the cells in a culture. The time required to kill all the bacteria in a particular culture at a specified temperature. And this is important for sterilization procedures. Heat treatment is simple, inexpensive, and effective. It's the best method if the material being treated is not damaged by heat. Heat penetrates to kill microorganisms throughout the object. The use of heat is bactericidal. The resistance to heat depends on the population. Factors that affect sterilization by heat would be time, the organism, temperature, concentration, presence of organic material, pH, spores or no spores, type of heat, and osmotic pressure. Moist heat destroys microorganisms mainly by denaturing proteins. So that leads to a coagulation of proteins. So here we have a great tertiary structures of proteins, and when we heat them, we cause them to denature, and then what ends up happening is they begin to coagulate with one another. Example of moist heat would be boiling water at 100 degrees Celsius. Usually, we want to boil water to kill vegetative cells for 10 minutes. So boiling water destroys the vegetative cells of most bacteria, but remember, they're not effective in killing spores. The only thing effective in killing endospores is an autoclave. It only inactivates some viruses. So boiling water doesn't kill all the viruses, and it's definitely not going to kill endospores. Another type of moist heat is the autoclave. The autoclave is a machine that uses steam under pressure. Raising the pressure in the chamber raises the temperature of the steam. So you can get to about 121 degrees Celsius at 15 psi. So psi would be pressure per square inch. So what you can see here is we have a autoclave, and it's exactly how it looks in the lab. It's in the back, and then we put all the stuff onto this shelf, and then we close the door, and we set the timer, and it will autoclave for us. Of course, the autoclave can kill endospores and hepatitis, and it can do so in 15 minutes. Volume versus time. The larger the volume, the higher the sterilization times. So please take a look at Table 4 on page 100 of your textbook. Autoclaving is the most popular sterilization technique in the laboratory. Another example of moist heat is pasteurization. Where does the name pasteurization come from? If you said Louis Pasteur, you are correct. Pasteurization was a process invented by Pasteur to destroy organisms without affecting the flavor. Pasteurization reduces spoilage organisms, and pathogens. We do know that there are some thermoduric or heat-resistant organisms that will survive. It will kill the food pathogens without affecting the flavor. There are different pasteurization times and temperatures for different products. For example, we have to take a look at the fat content. The more viscous the substances, the more protection it provides for microorganisms. So if we remember what viscosity is or what we mean by viscous, water is not that viscous, whereas syrup is very viscous. So milk, if it's very thick, will have to probably pasteurize for a much longer amount of time. The phosphatase inactivation test. This test determines whether products have been pasteurized or not. There is an enzyme called phosphatase that exists naturally in milk. If phosphatase is inactivated, the product has been pasteurized. So we know that phosphatase is a protein, it's an enzyme, and if we expose it to high temperatures, it should denature and not work. So if we inactivate it, then we know that we heated it enough for the bacteria to die. But if it's still activated, then we haven't heated it thoroughly. Equivalent treatments is 63 degrees Celsius for 30 minutes. High temperature pasteurization involves heating milk to 72 degrees for at least 15 seconds. Ultra high temperature pasteurization raises the temperature from 75 degrees to 140 degrees and then drops it back to 74 degrees in less than 5 seconds. We do ultra high temperature for products that do not need to be refrigerated. Dry heat sterilization kills by oxidation or burning. It is usually used to sterilize metal objects and glassware. Examples of dry heat is flaming, direct flaming. An open flame is a form of dry heat used to sterilize inoculation loops in the mouth of culture tubes. So this is what we do in the lab doing the aseptic technique. Hot air sterilization usually requires more time than moist heat because we know the heat does not transfer well in air. So we need about 170 degrees Celsius air maintained for 2 hours. So the heat does not transfer well in the air, so it requires a lot more time than moist heat. So if we take a look at the equivalent treatments, 15 minutes for the autoclave versus hot air for 2 hours, we think we're going to use the autoclave. So the hot air sterilization is not used very often in labs. Other methods of control: filtration. Filtration removes microbes, so it filters out or removes microbes. It is used to sterilize heat-sensitive materials such as enzymes and vaccines. We also use it to make heat-sensitive media. The size of the pores of a membrane filter is about 0.2 micrometers. So the size of the pores is about 0.2 micrometers. I'm sure you guys heard of HEPA before in filters. HEPA removes microbes that are greater than 0.3 micrometers. And membrane filtration removes microbes that are greater than 0.22 micrometers. Low temperatures inhibit microbial growth. So refrigeration slows the growth of many pathogens. So we're not killing them. We also can do deep freezing. Desiccation, or the absence of water, prevents metabolism. Microorganisms cannot grow or reproduce. Osmotic pressure causes plasmolysis. It's used to preserve foods in a process called curing. So we can use salt or sugar to preserve our foods. It resembles desiccation, in which water is going to want to leave the cell. Because remember, plasmolysis occurs when you're in a hypertonic solution. Water is leaving the cell. So we tend to use this on meats. Radiation damages DNA. There are two types of radiation: ionizing and non-ionizing, and they're based on wavelengths. Ionizing radiation, examples are X-rays, gamma rays, and electron beams. They have less than 1 nanometer of a wavelength, and because they have such a small wavelength, they have very high energy, which create hydroxyl radicals from water, which will damage organic molecules, especially DNA, by stealing electrons. It sterilizes medical supplies, some pharmaceuticals, and food preservation. Non-ionized radiation, or UV radiation, has a longer wavelength. So because it has a larger wavelength, it has less energy. It forms thymine dimers, which leads to DNA mutations and inhibition of replication. That low energy penetration means that it cannot penetrate. So it doesn't penetrate well. So in order to use UV to kill microbes, direct exposure is required. So we use this for vaccine disinfectants and germicidal lamps in hospitals. Microwaves kill by heat, but they're not especially antimicrobial. So the waves that the microwave produces does not kill. It's the heat that does the killing. Here ends part A of chapter 4.